we're going to talk quickly about DNA replication. I want to try and keep this video fairly short so that you guys can continue with your readings. But let's start with, when does DNA replication occur? So this is the cell cycle. As cells grow and multiply, they go through this cycle over and over again until cells start to die and they exit the cycle. For the first part, there's just cell growth. The individual cell grows. Second part of the cycle is the replication of DNA, and that's what we're going to focus on today. Later, there's more growth that happens, and then finally mitosis, which you've learned in standard biology. We also have different types of chromosomes for prokaryotic cells, which are circular. If you follow this all the way around, it's circular, very simple, free-floating in the bacteria cell. In eukaryotic cells, we have our DNA, which is wrapped around these histones, a group of 8 histones, and then those histones, which are just a protein, form into rings and finally into bigger and bigger chromosomes. And all of that is stored within the nucleus of the cell. For our replication, we need to have a place of an origin of replication. Prokaryotic cells, they just have one, and it goes around and around and around, and this side goes around and around and around until the whole thing is complete. For eukaryotics, we have multiple sites of origin, boom, 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 and the cell will, or the DNA will replicate at those simultaneously, similar to the way this is. So here's our prokaryotic cell. It's replicating by splitting, and it goes all the way down until there are 2 rings. Then the cell will divide. For eukaryotic, there are little bubbles, and those bubbles will enlarge, enlarge, enlarge until they form 2 completely new strands. So let's get into the details about it. I've gone over this briefly in class, but the first step is helicases, which are a protein, start to unwind the helix and start to break it apart. You've got the single strand binding proteins, which stabilize the DNA because it really wants to coil back up, and if those guys aren't there, then it's going to just wrap back up. The third thing that happens is there has to have a primer. It's not shown here, but there always has to be a RNA primer, and it's just a couple of bases that is put there by a primase. Once it's there, replication can start happening by DNA polymerase. For the leading strand, it can go continuously from 5' to the 3', because that's the way it's able to do it. Like, it doesn't need to double back on itself. For the lagging strand, which is very frustrating, we have to put the DNA primer there, and then it'll go through and do a little bit until another primer is needed. Those little fragments of DNA, after they're all put back together, are called Okazaki fragments. And those Okazaki fragments, they need to be stitched together. So that's what the job of this DNA ligase. He comes along and he reads the DNA, and any break that he has, he's going to fix and stitch it together. That would be my daughter crying in the background. So those Okazaki fragments, once they're all stitched together, that's a full strand of DNA. And it's going to do this over the entire thing. The overall direction of replication is towards the replication fork. Again, you have to go 5' to 3'. So on the lagging strand, it's very difficult. You got to do it step by step by step. For the leading strand, it's much easier because it's already going 5' to 3'.